Okay, well, welcome everybody. This is Clinical Trial 101. This is the first in a series of that will be happening, hosted by the clinical trials team. They'll be doing um, uh, every uh, every month. They'll be hosting one, switching from special topics to having drug sponsors present. Um, I just want to thank both Jill and Rob for presenting this. They're both members of the I AM ALS clinical trials team. This is a team that meets weekly on Thursdays at 1 p.m. Eastern time. And if you're interested in joining them, feel free to shoot me an email at michael@iamals.org. Their mission is the clinical trial team partners with researchers, government agencies, and drug sponsors to ensure access, efficiency, and humaneness of ALS therapy development and approval. It collects and distributes information regarding clinical trials to the ALS community. And a little bit about I AM ALS before we get started is we are a patient-led community that provides critical support and resources to those living with ALS, their caregivers, and loved ones. This includes things from our community support team, including support groups and the peer support initiative, which matches mentors to mentees. Revolutionizes how we cure ALS by empowering and mobilizing advocates to raise awareness of the disease, and engages with policymakers to drive the development of cures. I AM ALS is going to continue to amplify the voices of people affected by ALS to fight for change until ALS is eradicated. Just a few items before we get started is I'm going to ask you to mute yourself if you're not speaking. If you have a question, feel free to raise your hand, type in chat. I'll be reading questions that are typed in chat. But if you have a question anytime during the presentation, feel free to let us know. You can either raise your hand or also vocally say something or enter in the chat. I'll be monitoring that. But now I will turn it over to Jill and Rob.
Thanks, Michael. Great to have everyone here tonight. We want to welcome you to Clinical Trials 101. Just a quick disclaimer, this webinar is for information only. We are not recommending any specific trial, and we are not really going to talk about specific trials. We're going to give you an overview of how clinical trials work and some things that you may not be aware of. So here's our agenda for tonight. And again, as Michael said, if you have any questions, please put them in the chat and we'll answer them as we go along. We are a very informal group here tonight. And we'll also have a question and answer at the end of the presentation. So what we're going to do is talk about clinical trials, an overview, terms that you may need to know, the different phases of clinical trials, why people participate in clinical trials, the process of biomedical research and development. Sounds like a fancy term, but it's just clinical trials. The challenges that we've seen in ALS clinical trials, and some innovations that you may not be aware of. And certainly, as I said before, if you have any questions, we'll talk about those at the end of the presentation or during. So what is a clinical trial? Well, we have this nifty definition from the FDA that says clinical trials are research studies conducted with human volunteers and designed to answer specific questions about safety or effectiveness of drugs, vaccines, other therapies, or new ways of using existing treatments. A couple things to highlight. As we see here, it says human volunteers. When you hear about things going on in a lab or things with animals, those are preclinical trials. So we're focusing on human volunteer clinical trials. Also, it's important to note that clinical trials are a necessary part of the drug development process. The FDA, or Food and Drug Administration, will not approve a drug without them. So tonight we're going to talk about two types of clinical trials, interventional and observational. Both start with a hypothesis. Both have a sponsor, which can be a drug company, a research institution, or a single investigator. And in an interventional trial, participants will be in one of two groups. The first group is one that receives an experimental drug or therapy or procedure. The second group would be part of the control or placebo group, we call it, that receives a placebo or an inactive substance. We have a link on the slide here to I AM ALS's signal dashboard, and it's a great resource if you're looking for any disease-modifying interventional trials. So the other trial I spoke of is observational. The biggest distinction here is that participants are observed and information is collected. So there's no intervention and, of course, no placebo group. We've noted a couple observational trials, one, the pre-symptomatic gene carriers, and also a natural history study. Again, just collecting information and observing the participants in these trials. A really good example of an observational trial that you might have heard of is ALS TDI's ARC study. Again, we have a link on the slide here if you're interested in learning more about that or even participating. So, why do we even need clinical trials? Well, we know that people living with ALS tend to focus on trials of treatments that can slow, halt, or reverse disease progression. But it's really important to note that clinical trials are advancing medical research and improving healthcare, leading to better prevention, diagnosis, and treatment. All clinical trials are important, even the ones that fail. While it's frustrating to be part of a failed trial, the information gained from that trial helps bring us closer to an answer for ALS. Now let's go over a few terms. You've probably run across these, or if you haven't, you're going to run across them in the clinical trial world. And I'm just going to go down the list here. So the first one is randomized. And this is when individuals are assigned arbitrarily to receive either the experimental treatment or the placebo. The next one is double-blind. This is where neither the researcher or the participants know who is receiving the intervention and who is receiving the placebo. So I guess the next best term to define is placebo. And as mentioned above, this is an inactive substance that looks the same as the active treatment and is given in the same way. And placebos are necessary to determine if the experimental therapy is having any effect. People in the placebo group are monitored and data is collected on them, just like the people with active treatment. Next term is placebo ratio. A percentage of the trial's participants receiving active investigational treatment and placebo go through randomization. Typically, the ratio is 1-to-1. So one person is going to get the active treatment and one person is going to receive placebo. There are other ratios. For instance, the Healy platform trial has a ratio of 3-to-1. So 3 people receive the active treatment and one person, again, receives a placebo. Next is control group. The term used for the group of individuals who are not receiving the active treatment being investigated in the trial, or sometimes we call this the placebo group. Protocol. This is the overall plan of the clinical trial. So it includes the duration of the trial, the number of participants, as well as the endpoints and other information. So let's talk about endpoints. This refers to what is being studied during the clinical trial. Endpoints are decided before the trial begins and are measured at the end. They are used to determine if the therapy works. Examples of endpoints would be a rate of progression measured by the ALS-FRS-R scale, rate of survival, the rate of FVC or forced vital capacity, and SVC or slow vital capacity. Finally, on this slide, we have the ALS-FRS-R. This is the scale that measures 12 aspects of physical function, ranging from the ability to swallow and use utensils all the way to the ability to climb stairs and breathing. Okay, another group, hang in there with me. Next is the principal investigator, and this refers to the lead person at a medical institution that is running the clinical trial. IRB, which is the Institutional Review Board. This is a group of doctors, scientists, and lay people who review the research plan before the trial starts to ensure participants' rights, safety, and welfare. This group is one of a couple groups that are looking out for the participants' best interest. Informed consent. This is a document that details the trial purpose, the length, the individual's roles, testing requirements, number of visits, potential risks, side effects, benefits, and confidentiality. You're going to receive this document before you start a clinical trial, and it is extremely important that you read it and understand it. It's very comprehensive. It'll tell you everything you need to know, maybe some things you don't want to know, but again, you're going to sign this document, so please take the time to read through it. The Drug and Safety Monitoring Board is an independent group who monitor trials on an ongoing basis for, again, participant safety and data integrity. Open label extension of a trial wherein all participants, including those who are in that placebo group that we talked about, are offered the opportunity to take the active drug. So if you were receiving the drug and want to continue to do so, you can do that. And also, as I mentioned, if you were in that placebo group or control group and would like to receive the active drug, you can do that. Expanded access, we've all heard about this through the Act for ALS. Expanded access is a program for individuals who are ineligible for a clinical trial. And if you did not meet the inclusion criteria, or you met some of the exclusion criteria, you couldn't participate in that clinical trial. But expanded access gives you an avenue to get that investigational therapy. Last but definitely not least is the FDA, the abbreviation for the U.S. Food and Drug Administration, which is the government body responsible for ensuring that drugs are safe and effective before making them broadly available. That's a lot of terms, but again, you'll run across them every now and then, and it's good to know what they mean. Now I'm going to send you over to Rob, and he's going to give you a more specific view on the clinical trial process. Rob?
Thanks, Jill. Appreciate that. And yeah, these terms will come up as I go through my part of the presentation as well. So buckle up. We're going to start off with a light topic, starting off with statistics. I noticed that was absent from the agenda, which is probably a good idea because people would have probably dropped off before we even got started. So anyway, we're here and we're going to do statistics. I'll try to make this as painless as possible. I'm not going to go into a lot of depth. It's not going to be a comprehensive description of statistics. I'm only going to give you what you need to kind of understand what we're talking about here, and hopefully understand when you see clinical trial results, what that all means. We're going to start with the end, and that's the p-value. So that's kind of the measure of the success of a clinical trial. And what it means and what it refers to is the probability that a result, in this case, let's talk about the difference between the treatment group, the people receiving the treatment, and the placebo group. If you see a difference, what is the probability that result, that difference occurred by chance? And so in the world of statistics, a p-value of less than 0.05 has been sort of regarded as the acceptable definition of something that say, oh, this didn't occur by chance. This is actually a real difference between groups. The FDA has adopted that. Other statisticians have adopted it as well. And what does that mean? It means that there's a 5% chance that it occurred by, or 5% probability that it occurred by chance, or a 1 in 20 probability that it occurred by chance, or a 1 in 20 chance of a false positive. So what goes into that p-value? Well, there's effect size, and that's the magnitude of the differences between groups. And I'll give you an ALS example. Let's say there's a treatment that, on average, in the treatment group, reverses disease progression. So actually, functional rating scores improve. And then in the placebo group, you see what we normally see, unfortunately, in ALS is a decline in function. Well, that could end up in a pretty significant treatment or magnitude of difference in a large effect size. Normally what we see is a smaller effect size where you might just slow progression, but progression occurs in both the treatment and the placebo group, even with a successful drug. Next up is standard deviation, and that describes the variability between measurements or between the participants in a trial. For example, as we know, ALS is a very heterogeneous disease, and so there's a lot of variability in progression rates among individuals. And so that leads to a pretty high standard deviation relative to other disease states potentially. And then also there's finally is N. So that just refers to the number of participants or observations. So all of those things go into the p-value. And then next up is this process of biomedical research. Now, this was in the agenda. And so we're going to kind of walk through this, starting as Jill described the preclinical phase. This is the work that occurs in the laboratory, in the dish, in animals, every place but not in people. So a clinical trial, again, is as Jill said, is defined by testing a therapeutic agent or testing a therapy in people. So it starts with phase 1, and as you can see, they get larger in terms of the number of participants as you go along. I've seen this slide a lot of times. I spent my career in the pharmaceutical industry. Every time I see it or something like it, I say to myself, yeah, but, and so this is intended to be a general description. All of this is a general description, and there's exceptions to that. And we're actually going to talk about one of the exceptions that's part of our recent history in the ALS world. So anyway, starting with phase 1, those are the smallest ones, and they're less focused on efficacy. They've been described as non-therapeutic. So they are more focused on the safety and what happens to a drug in the body. And so they're smaller, fewer participants, they tend to be shorter. If things look good in phase 1, it's safe. I mean, this is the first time they've been tested in humans, so that's obviously an important focus. If it's safe and does what's expected, then it can move on to phase 2. This has been described as an exploratory phase. So it's a larger trial. This is the first time often that they're looking at efficacy, and they're comparing the treatment group to a placebo. So it's a clinical trial. They have placebo and treatment groups, but they are it's focused also on efficacy and now and also on safety. But it is exploratory, and so they're not intended to be registration enabling. So generally, the data from a phase 2 trial isn't the last step and doesn't go to the FDA to review. But what it does do is generate important information for the design of the phase 3 trial. The phase 3 trial is described as confirmatory. So in other words, that trial is the one that provides the definitive data on efficacy and safety that's needed for the FDA approval. So once the phase 3 trial or trials conclude, the data go to the FDA, and presuming they're successful, the data goes to the FDA, that gets reviewed, and hopefully the drug gets approved and is available for general use. Sometimes there's a commitment and an expectation that the drug sponsor is going to do additional work post-approval, and these are called phase 4 trials. But we're going to focus on phase 1 through 3 in this presentation. Now, as you might imagine, this is a lengthy process, and an average of 10 years has been quoted as common. So we're going to dig a little bit deeper into the trials. Yeah, go ahead.
So one is I'll give them to you at the same time. You can choose how to tackle them. One is Paul says, I got on late but didn't see the term biomarkers. So if we could explore that term. And then Robin asked the question, are the same people involved in all phases?
Okay, I'll start. I can take those in order, and if anybody else wants to jump in. So yeah, Paul, biomarker is something that is a way of measuring something in a clinical trial. The one that I guess the endpoint that gets used most commonly is the ALS FRSR scale, which is the functional rating scale. It's not really, considered a biomarker. An example of a biomarker, and unfortunately we don't have as good of ones in ALS as they do in other diseases, would be, let's say, hey, does this drug lower cholesterol? Well, there's a pretty easy blood test for that, and you can, you'll be able to see quickly whether or not a drug is actually lowering cholesterol. We are, the community and the scientific community is exploring other biomarkers like neurofilament light. There's digital biomarkers like accelerometers that you can wear. There are a number of them, but unfortunately, none of them are considered by the FDA as something that can be used for registration. So they're not registration quality, but hopefully we'll get there in the not too distant future. And then, Robin, to your question, are the same people involved in all phases? Generally, no. So I think that they'll recruit a new group of people, and it could be confounding to the results. Let's say you were in a phase 2 trial, and you receive the active treatment, but then end up in the placebo group of a phase 3 trial. That could throw off the results. So in general, no, they're different people every time are part of these. There is a notable example coming up here that I'll share with you though. All right, digging in on the phase 1, again, safety and dosing. They're shorter in length, several months, and sometimes they're even just a single dose. In a lot of cases, you just get a single dose. They're smaller than the other 2 phases. Less than 100 is, sometimes it's 1. And so back to that N of 1. And they might just be looking at, what is the reaction that a person has to this treatment, and how does the body react to it? What is and where does it, how's it metabolized, and how does it get through the body, and does it reach the spinal fluid? And you can do that with a very few number of patients. A lot of times they're not ALS patients at all. They're healthy volunteers. So they're the participants, vary very broadly. Commonly, they do move on to the next phase. So hopefully there's been enough preclinical work to say that there's a good probability of success. They're not inexpensive. So you might think that if they're not that long and there are not that many participants, it's going to be inexpensive, but it's not. And then you may see trials described as 1A and 1B. Maybe 1A is healthy volunteers, 1B is people with the disease. I like to think of it just sort of at 2 sides of phase 1. Phase 1B is more like a phase 2 trial, and phase 1A is almost more like preclinical in a way. Let's move on to phase 2. And this is the exploratory one, as we described, and it's hypothesis generating. So this helps design the phase 3 trial. They're longer. They include efficacy, so they're looking at efficacy, but then of course side effects and safety, that's an important part of it as well. They're longer, so several months to a couple of years. There are more participants. There are several hundred people. Fewer of them progress on to the phase 3, and they get more expensive. And again, there are also 2A and 2B. 2A is a little, is more like a phase 1, and 2B is more like a phase 3. So I hope that makes sense. And to bring this home, I thought it would make sense to bring in an example from our recent history in the ALS world, and that's the Relyvrio trial, the Centaur trial, done by Amylyx Pharmaceuticals on the drug Relyvrio. So that one had an N of 137, so there was 137 participants. That includes both the active and the placebo. And the actual result of the trial was that the people in the treatment group had a slower progression by 2.32 points on the functional rating scale. So back to the statistics, that's the effect size, and it was a pretty significant effect size. And they had a 2-to-1 active to placebo ratio, so that was considered humane, and we like to see those more. The ratios skew more to the active treatment, and the p-value was very significant. So it was a 0.03, which means there was only a 3% probability that this positive result occurred by chance. So Amylyx Pharmaceuticals, with just phase 2 data, now we said the phase 3 is generally necessary for applying for approval. Amylyx, with a lot of help from the advocacy community, convinced the FDA to review their submission, and they got approval, conditional approval, but they got approval for their drug based upon this very positive phase 2 data, under the condition that they conduct a larger phase 3 trial. So let's go on to phase 3. So again, phase 3 is the confirmatory one. It's focused on efficacy, but also you have to monitor adverse events or safety, of course. This can be 1 to multiple years. So this is a longer trial, several hundred to thousands of people. Now, ALS is going to fall on the lower end of that. And the number of treatments moving on to the next phase is lower, which is kind of registration, and the cost is more expensive, as you might expect for a longer trial with more participants. So it's interesting to note that in general, the FDA requires for, this is not just for ALS, but for all diseases, 2 adequately and well-controlled phase 3 trials as a condition for approval. I think with ALS, they're likely to exercise regulatory flexibility and approve a drug with maybe just 1 trial, as long as it definitively shows results. Again, back to statistics, a p-value of 0.05 or less.
We have another question.
Mm-hmm.
Is it possible to run 2 phases simultaneously, such as phase 2-3? I have a special surprise at the end of this that will answer that question, if you'll allow me. Sounds good.
Okay. So let's go back to Relyvrio. This is the sad part of the story. The phase 2 was a very exciting part. This is the sad part of the story. So Amylyx very confidently designed their phase 3 trial with every expectation that it would work. The FDA agreed that as long as they conducted this trial, that they would approve Relyvrio, known as Albrioza in Canada. They did, and it was available to everybody with ALS. And so they conducted a phase 3 trial. This one was called Phoenix. They had 600 participants. They had a 3-to-2 active to placebo ratio. And so looking at the statistics, looking at the effect size from phase 2, this should have been more than enough to confirm that Relyvrio was an effective drug. It turns out it was not. So this was a 48-week trial, and the difference between the 2 groups was only 0.34 points on the functional rating scale, which was not enough to show a statistical difference. So the p-value on this one was 0.667, which means that there was a 66% chance, or 2/3 chance, that this result actually occurred randomly. So there's a 66% chance that it was a false positive. So this is clearly not sufficient for regulatory approval, and unfortunately, they had to remove the drug from the market. So it's no longer available to new patients. But it's interesting, and I'm still sort of puzzled how this happened. Was the phase 2 trial just in that 3% probability that it was occurred by chance? I find that hard to believe. I know that people are going to be investigating the data for this for some time to come to try to figure out why this happened and hopefully prevent it from ever happening again. So on to something else with that behind us. Why do people participate in clinical trials? I like to start with this. Participation is an act of altruism. I hold people that participate in clinical trials in the highest regard. because they advance medical research, and we need that in this community. We need that for this disease. We need to improve our knowledge of the disease so that we can get closer to slowing progression and curing this disease. Trials are necessary. So participating in a trial helps get drugs to market. So that is a key reason for doing this. But then as you're sitting there in front of your neurologist, you're wondering, what happens? Is it possible? And I know this happens because we all think this way. Is there a possibility that I will get this experimental treatment and it will be beneficial? It will slow my progression. It'll halt or reverse my progression. And that is another great reason to participate in a clinical trial. But that comes with caveats, and you should know that in a placebo-controlled trial, there's a possibility that you're going to get a placebo. If it's a single dose trial, there's very little probability that you're going to receive a benefit from that. Keep in mind that it's an experimental treatment and may actually be harmful. Again, we're studying safety. We're monitoring these side effects. We don't know. So again, it's an act of altruism. It's an experimental treatment. It's an experiment. But I want to say thank you personally on behalf of the entire ALS community. If you have participated, if you are participating, if you do participate, thank you from the bottom of our hearts.
We have another question.
Yeah.
Are there any key ALS trials that you think would be good to keep an eye on?
There are plenty. Unfortunately, a lot of them are now in the phase where they're analyzing the data. I encourage you to follow up with us afterwards. I encourage you to go to I AM ALS's ALS Signal, which is you can filter that down by clinical trials. And then also another great resource is ALS TDI's Clinical Trial Navigator. That's a newly available tool that's quite helpful in finding things that might be appropriate for you. And to recommend something based upon whether or not I think it's going to be successful, again, these are experiments, and as Relivrio has shown us, we don't know whether they're going to be successful. So I personally try to shy away from recommending something based upon whether it's going to be beneficial to you. But I will say that there are tools for helping you find trials. And I also will say that it's very important to do that and to advocate for yourself. Okay, so you've decided that you'd like to participate in a clinical trial, and you start looking at, as we talked about, finding clinical trials that might be appropriate for you. It's important to know that there are eligibility criteria that you need to know about before you can participate in a trial. They're usually broken down into inclusion and exclusion criteria. And as you might imagine, depending on how they're worded, it could be one or the other. But I'll give you a couple of examples to get us started. An inclusion criteria is something that you must have, a characteristic that you must have before you can participate. So if let's say it's an efficacy trial, then they're testing a drug for ALS, then you need to have a confirmed diagnosis of ALS. If it's an exclusion criteria, these are things that prevent you from participating in many trials because they're investigational drugs. They may not know, if you're pregnant, you can't participate for a variety of reasons. So while excluding people from trials may seem counterintuitive and maybe even cruel, there's an important reason for doing it is to create a group of individuals that are likely to provide definitive results. Now, think back to the statistics. One of the things that you want to do is have as homogeneous a group as possible while still being broadly inclusive. And the homogeneity helps create a lower standard deviation so that you have a higher probability of achieving that gold standard of a 0.05 p-value. So some examples of inclusion and exclusion trials or in ALS trials, time from diagnosis or symptom onset. So if it's been too long since your symptoms started, you may be ineligible for a trial. If your breathing score is inadequate and your breathing is not good enough, you might be ineligible for a trial. Some trials will require you to have a minimum ALS FRSR score. So, it has to be high enough so that there's something to measure in terms of disease progression. Ability to swallow. You may wonder, well, if it's an oral drug, why can't I just use a feeding tube? Well, drugs are absorbed differently, whether they're taken by mouth or by feeding tube. And then also if it's an extended release, for example, it may not work the same way if it's crushed and delivered by a feeding tube. Many are, and we'd like to see more trials that will allow that. Another thing to consider is participation in another ALS trial. So if you're receiving an investigational treatment, more often than not, you're going to be prevented from starting another one. You can't generally be in 2 clinical trials at one time, just because it could confound the results pretty substantially. And then finally, absence of tracheostomy. Generally, if you have a tracheostomy, you're not likely to be included in a trial. All right, so what does the process look like? So it starts with discussing this with your physician. Am I eligible for this? Do I meet generally the inclusion/exclusion criteria? Is it something that you're interested in doing? And if it is, and go to the next step, Jill talked about this, the informed consent, and I can't emphasize this enough. This is not one of those waivers that you sign as when you download a new app on your phone. You should read this one. And you should make sure that you understand it. And if you don't understand it, make sure you ask questions of people that do understand it so that you know what you're getting yourself into. There may be things that you don't know that you're additional screening. So that would be the next part of it. And let's say all of that works out and you are eligible for the trial and you're still willing to participate after signing the informed consent, then you are assigned randomly to either the treatment group or the control group. In theory, you shouldn't know, and that's why they disguise the placebo so that you don't know. Your physician doesn't know. Nobody knows except for once they complete the unblinding. So nobody really knows whether you're in the active group or the control group. So you receive the medication and you'll visit either in clinic or telehealth visits throughout the duration of the trial. So they'll be collecting data while that's going on. And it's important to note as you turn the corner here and during that time, you can still visit with your regular physician. So that's, it may be a different and likely will be the investigator for the trial, and who you're seeing for your clinic visits or your clinical trial may be and is likely to be somebody that's not your regular physician, but you should continue the visits with your regular physician. And then once that's done, the study concludes. As Jill talked about, sometimes there's an open label extension. We like to see that as often as possible to have an open label extension. And that means that the placebo-controlled portion of the trial ends, but then everybody, both people that were on active treatment and people who are on placebo, now convert over to active treatment. And that is when the data analysis starts, but you can, if it's in an open label, you can still be on, you'll still be on the drug. And then there's the excruciatingly lengthy, oftentimes, component, which is where the data gets analyzed before the results get published. All right, so as I've been around clinical trials in ALS and other disease states for a long time, and there are certain things that I've come across in ALS trials that make them more challenging or certainly different from other clinical trials. So first one is the functional rating scale, ALS FRSR, and that is, we really wish there was a better way to measure disease progression. Unfortunately, there's certainly not one that's accepted by the FDA at this moment. So that the that scale and survival are really the two things that the FDA will accept as evidence that an ALS drug is effective. It's a subjective trial. It tends not to be super sensitive in certain cases. So there's a lot of issues with it, and there's a lot of people actively looking at alternatives. We talked about this too, the disease heterogeneity. That is, again, back to that standard deviation that makes it hard to achieve your 0.05 p-value. And then we also talked about this too. There are no accepted biomarkers to measure disease progression. Like I said, neurofilament light, there's hope there, and some others, but there's not one that could replace the functional rating scale as a way to measure the success of a drug. Another one, and this one I really struggle with. I think we have to do better here. So the time between symptom onset and ALS diagnosis is long because it's a diagnosis of exclusion. You have to rule everything else out. You have to find a neurologist that's thinking ALS before you can get diagnosed. And so a lot of times, a trial will say, well, you can't be in the trial if it's been this long since symptom onset. Well, you may not have had a diagnosis even before then, so you wouldn't even have had ALS trials on your radar. So this is a challenge, and it on to the next point, it reduces the size of eligible patients. Well, ALS is not an ultra rare disease, but it is a rare-ish disease. It's not, hypertension or hyperlipidemia. It is, there's not as many patients. As we know, there's a lot of them, but it's harder to achieve that N, especially since a lot of patients are ineligible by the time they're diagnosed. As ALS patients know, and caregivers know, and those of us in the ALS community know, it's difficult to travel as mobility becomes challenged. So there are travel challenges, and there are geographic barriers. There are trials that are occurring in Europe that they're not in the US. So there's geographic barriers. And then finally, lack of awareness. And this is where I want to talk about advocating for yourself. So I've heard way too many stories about people that are diagnosed with ALS, and they talk to their neurologist, and the neurologist says, I'm not aware of any clinical trials. Well, there are clinical trials out there. They may not be in their facility that they're operating out of. And so there is a lack of awareness both among the patients and then the physicians. So again, it's important to advocate for yourself. It's important to do the research or get somebody to help you with the research and find out what's available so that we can, kind of shrink this awareness problem. All right, so on to innovation in clinical trials. I like to end with bring home a bright spot here. So we are seeing, because ALS is particularly challenging, we're seeing innovation in ALS clinical trials. And it's hard to run just a kind of a traditional trial because of the challenge that we've seen. So the platform trial structure is being done out of Mass General. And so this is a protocol that that does a lot of great things. One of the things is it can help reduce the time and the cost. Because everything is under a single master protocol, you can drop drugs in and they can all be tested simultaneously. So it's not tested on the same people, but because they don't have to start over again with a new trial, they can just put drugs in. So the way the Healy trial worked is they started off with 4 regimens, A, B, C, and D, and they were running simultaneously. So they were recruiting patients for all of those at the same time. Then they added regimen E, and then regimen F, and then regimen G. So one of the cool things that you can do with this is because it's all being done on the same protocol, all the patients are being recruited using the same criterion. They can use the placebo across these groups. So if you're recruited into this, you have a 3-to-1 chance of getting the active treatment. So that's, we really applaud that. I think that's great. And again, it saves time and money. Unfortunately, so I think regimens A through E have read data. None of them have been definitive. Some of them are moving on to additional trials, which is good. Some of them have shown no benefit, and so unfortunately they're not going to continue. Regimen F and G, we don't know the answers to those yet. We don't know how they did. However, they have completed recruiting, which is good, because now we will find out whether or not they worked, which is bad if you are looking for a clinical trial. These are no longer looking for new patients. All right, so I promised you this one. Is there a way to combine all the phases into a single protocol? And it turns out that there is, and Ionis Pharmaceuticals has done this. There is the, I can't pronounce this name. I've tried, and I cannot get it to come out of my mouth. So I'm going to go with Ion 363 for FUS gene carriers. This, if ALS is a rare disease, this is an exceedingly rare gene variant that is, that is a very small subset of the ALS patients. So this drug, they've had a very, and it would take probably a whole other webinar to describe this protocol, but they've been able to combine it all into a single protocol, and patients will move from phase to phase to phase as they continue to be studied in this. And so this is innovative. It can reduce time and the spin up of individual studies. So this is we're glad to see this, and this is a very important one. And then finally, basket trials. So a basket trial, and I've seen this a lot in oncology and other areas, is where you take a single drug and you look at it in a lot of different disease states. It was more common in cancer because, there's, I think, believed that there was more commonality in cancer than there is in neurodegenerative diseases, but there is a trial going on right now with baricitinib that's being tested in ALS and other, potentially related conditions like Alzheimer's, et cetera. So I'll stop there. Are there any other questions coming out of the chat?
We had one question, which was Mary said, regimen G was already full. Will there be an H? And Diane responded with, we haven't heard specifically, but it's likely. Researchers and drug companies like the benefits of the platform.
Yeah, I can echo that, and I know that there's a lot of consideration, kind of ongoing with the Healy platform trial, and there's always folks that are, looking at it, and they would like to. The folks at Mass General would definitely love to get more people into that trial, as would we, as an ALS community, just because we love the design, we love how fast it can go, and we love what we can learn from that. So, anyway. Final slide here. We talked about expanded access before. We're going to close on that, and it'll be a teaser for an upcoming webinar. It's an in-depth topic. There's a lot of information, and just so you know, again, it's not technically a clinical trial, but you can find them in clinicaltrials.gov. So if you're looking for them, you can learn about them there. There are a couple of criteria here that it's worth noting. So it has to be an expanded access in general has to be for a serious or immediately life-threatening disease or condition. Well, certainly ALS qualifies. There has to be no comparable or satisfactory options available. Again, that's a good descriptor of the ALS, therapeutic options. And then finally, you cannot be eligible for a clinical trial in ALS. So this is a way to get access to an investigational drug if you're not eligible. So it's important to note that participating in an expanded access program or protocol should in no way prevent participation. Should not limit the available pool of people that would be in a clinical trial. Why is that? Why are we even saying that? Well, because there's no placebo in expanded access. So you might prefer to be in expanded access, but if you are in fact eligible for a clinical trial, you can't be in expanded access. I know that's a lot to contemplate, and I probably said more than I needed to or should, but again, stay tuned for a future webinar on that. And so with that teaser, I will take it to this last slide, which thanks you for your participation, and thanks, all of the people that, again, have been in or are in or will contribute by participating in the clinical trial. And what questions do people have? I also want to say thanks to my co-presenter Jill. I want to thanks to I AM ALS for hosting this, and thanks to all the people that actually contributed to this. It was an amazing group of people that helped put this program together.
And I just want to echo that thanks that both Jill and Rob and the people who helped out are incredible advocates and do the kind of advocacy that I love, which is people helping each other and people advancing the cause together. So thank you all for your incredible work. We do have some things popping up in the chat, so I'm just going to read them out loud. This is from Matt's iPhone, and Matt writes, "This is great. You mentioned the FDA only formally accepts ALSFRS score data and survivability data to assess if a drug works. What does it take for the FDA to accept other new measures that may come in the future, such as new biomarkers, digital biomarkers being studied?"
So I can take that, Jill, unless you want to. So it really comes down to solid evidence that the biomarker is highly correlated with something that they accept, and that it is related to something that we care about, which is slowing disease progression or survival. So it has to, and the way you do that is to study it, essentially. And we encourage all of our, the drug sponsors that we're in touch with to add biomarkers, whether they're digital biomarkers or biofluid markers, anything that we think and they think might be helpful and correlate with the most important thing, which is, like the clinical endpoint, which is either slowing disease progression or survival. So the more information we get on that, the more likely they are to accept that as a primary indicator, primary endpoint for approvability. I'll give you an example from my past. I used to work in the hypertension or high blood pressure space. The FDA is not super, I guess, amenable to change. And so for the longest time, we knew that, how your blood pressure is measured in two different ways. Like there's the high number and the low number. Well, for a long time, the FDA only accepted the low number and reductions in the low number as evidence that a drug was working, when we knew that the high number was more correlated with cardiovascular problems. It took a very long time for the FDA to accept the high number, the 120 and the 120 over 80, as evidence that a drug worked. So they're slow, but we're pushing them. We want to make sure that they understand that the functional rating scale ALSFRS-R is a blunt instrument, and that there are, hopefully better things that we can, learn about and use going forward. Neurofilament light probably holds the most promise, but there are others as well.
Yeah, I'll just second that, Rob, that there are groups that are working on a better scale than the ALSFRS, and there are many areas in this that need to be addressed, and they certainly are doing it. But as with anything with ALS, there are a lot of areas that need to be addressed. So it's good to have people in all of them.
Okay. Let's see. Mary asked this question. Thank you. Do I email the clinical trials myself when I find one? I might have missed one. And then Diane wrote, Mary, you can connect the company directly to learn more. they'll probably want to also speak with your neurologist to be sure the trial meets your needs.
I'll
And I have
just jump in
another.
and say,
Oh, go ahead.
sorry, I'll just jump in and say, Mary, also, if you go to I AM ALS's signal dashboard, then there, and you see a trial that you're interested in, there should be a link on that same line that you can email the company or the people that are putting on the trial for that.
Yeah, and just an additional layer of granularity to that, on that trial or on Signal, and then also on the ALS TDI trial navigator, there are links and to the individual sites that are participating in the trial. And so you can even find that on clinicaltrials.gov. So it should have the name of the contact person, an email, and a phone number that you can call. So clinicaltrials.gov has actually gotten a little better, and there's a map now of all the participating sites. So you should be able to then find something hopefully close to where you live, and then the contact information should be there.
Debbie just put the link for Signal in the chat, and I'll be emailing that out to folks too, along with TDI's new trial finder tool. One more question. Is the therapy that requires a monthly lumbar puncture to administer showing any positive results?
I'm not sure which one that is, but it's important to know that they usually don't start looking at the data, and they don't reveal the data until it's been fully analyzed. And so the trial has to be done, and then there has to be time for the data analysis to occur. So I'm not aware of anything right now that is, kind of in the readout phase that hasn't already read out, or is read out recently. But I'm happy to look into it if you can let me know which one it is. There's more than one that require a lumbar puncture.
Oh, Robin, you're muted.
Okay, my brother is currently receiving it once a month in Wilmington, North Carolina. And I don't think he's part of a clinical trial. I think this is a treatment that he's receiving.
Are you
Okay.
talking is it I still want to call it tofersen.
Oh,
That's it. That's the name.
tofersen, okay. Oh,
That's
yeah,
it.
that, I mean, Qalsody, yes. Oh, well, I mean, that was definitely a positive enough clinical trial that it was approved. So the data are meaningful, and I think that neurofilament light was actually a pretty reasonable part of that approval. And this was a good example, I think, of regulatory flexibility too, because they didn't do 2 large phase 3 trials. There was, but they weighed the evidence. And they also weighed the devastating nature of the disease and the limited options, especially for SOD1 patients. And so they approved the drug. But I think that based upon anecdotal data that I've heard, that it's actually been quite effective for people, and it's very exciting for the community.
But he may actually be getting as I see somebody else typed in the Qalsody, because he has the SOD1
Right.
genetic.
Yeah, tofersen is the generic name, and then once it went to market, they gave it a brand name, and the brand name for tofersen is Qalsody.
Okay. All right. Well, thank you.
Sure.
Any other questions? Well, very timely people. It's 7:01, so we only went over 1 minute. So I just want to thank the clinical trial team again for all the work. Thank all of you for coming and learning more about clinical trials. If there's anything in particular you'd like to know more about clinical trials, shoot myself an email at michael@iamals.org. And then the other thing is I'll be sending out those 2 links to you all later on, and this was recorded, so we'll have it in case you need to review this information again. But I hope all of you have a pleasant evening, and it was good to see some of you again and to see some new faces. Take care, everyone.
Bye. Thank you.
Bye.